I will shift to protein engineering for vaccine design. We work in the field of, as I told you before, in protein engineering for vaccine design. One of the main goals of our lab in the past has been to try to make a relationship between immunogenicity of protein and the protein structure. This fact is very well known in the model of immunogenicity related to molecular structure in the viral model. As you know, many people know, virus-like particles are particles devoid of the genetic material of virus, but they keep all the structure of a virus. And these virus-like particles are very immunogenic, and the immunogenicity of the proteins is completely related to the fact that the protein has to be organized at high level. The higher the level, the higher or the stronger the immunogenicity of the proteins. And this very simple fact has been exploited in the applications like the hepatitis B vaccine and the HPV vaccine for treatment of different diseases in human beings. But this, for example, is the HPV vaccine, and the vaccine is a virus-like particle that is highly immunogenic, and the cell protein and the capsule are less immunogenic. So the immunogenicity is related to the structure order. For vaccine engineering purposes, this is not applicable because most of the pathogens they do not form VLPs. So VLPs are being used as carrier to enhance the immunogenicity of weak antigens. And this is very simple. You can take, for example, a virus-like particle as a scaffold, as a protein scaffold, and use it to decorate with weak antigens. So these weak antigens will be presented to the immune system in a different setting, in a well-ordered structural setting, and they will become strong antigens. So this is a general idea of biotechnology, to use protein engineering for vaccine design. And I will very briefly present our model that was discovered and developed in our lab. And this model is based in a protein called BLS, that stands for Brucella lumacin synthase. And we have created a new platform for vaccine design based in this protein. This protein was discovered as a marker of active brucellosis. When I was doing my doctoral thesis, we were looking for antigens to differentiate human beings that have been infected by Brucella, that is a gram-negative bacteria that produce an anthropozoonosis. That is very important in our country. So we were able to discover by means of a monoclonal antibody an antigen that is capable of differentiate human beings that have the active disease compared to patients that have inactive disease. So we were prompted to study this protein, and we discovered that this protein has a 18 kDa apparent molecular mass, as you can see here, and has a sequence homology with lumacin synthases. What are lumacin synthases? Lumacin synthase is an enzyme that catalyzes the penultimate step in the biosynthesis of riboflavin, or vitamin B2. And this riboflavin gives rise to several flavocofactors like FMN and FAT that are very important for the function of many proteins. So this is an essential metabolic pathway in bacteria. So we did a lot of work trying to study the structure and function of lumacin synthases in Brucella. We discovered that Brucella has a new type of riboflavin lumacin synthase that is a decameric one compared with pentameric and icosahedric enzymes that have been previously discovered. So we did different work in this structure. we discovered that the protein is decameric, so it has 10 copies of this 18 kDa a small domain. As I told you before, it's a serological marker of active disease. We were able to demonstrate that this protein is highly immunogenic, both as the humoral and cellular arms of the immune response. The protein is highly immunogenic even in the absence of adjuvants and behaves as a potent DNA vaccine. The protein presents a very compact decameric structure that let us think that the protein has a highly protective effects because of the structure is very stable under different conditions. And this was the general idea that we're testing at our lab. For example, if you take a mouse and you administer only 10 micrograms of VLS in absence of adjuvant, you have incredibly high levels of antibodies through the whole life of the mice. That this is very important because most of the protein do not produce and do not elicit this type of antibody responses in the absence of adjuvants. We were able to demonstrate also that VLS is able to induce potent innate and adaptive immune response. In this case, we were able to demonstrate that the VLS via TLR4, this Tor-like receptor, is able to activate resting dendritic cells and stimulate them to production of different cytokines. And we know that also VLS is able to induce a strong in vivo cytotoxicity, which is TLR4 dependent. We know that the protein goes into vesicles, for example, here in macrophage, and these vesicles, you see a clear co-localization between TLR4 and our protein. So in some way, TLR4 is involved in these features, immunological features of our protein. So we developed an idea that the, immunogenicity can be related in some way with the molecular order. This is a kind of icosaedric lumacin synthase. In fact, this structure was solved in the 80s by Robert Hoover, that is present at this symposium, and this time was the higher molecular structure solved by X-ray crystallography. So we have this type of structure that we think, and I will try to convince in the next slide, that is an interesting scaffold for protein engineering for vaccine design. One of the main reasons is that if you see, in this case, I am showing you a pentamer of the decamer. If you see at the end terminus and you see the arrows, the 5 end terminus of this pentamer are having a different conformation in different monomers, clearly showing us that the end terminus of the protein has a large conformational freedom. So this will be a very good acceptor site for antigens, especially protein antigens, because we reasoned that we can put their antigens without compromising the stability and the structure, especially the quaternary structure of the protein. So what we designed at the lab was a cassette system to clone different antigens, simply by inserting here between 2 restriction enzymes any antigen of interest, and any antigen of interest will be produced as a monomer here, but the bacteria, in this case E. coli, will produce the decameric particle, and we will present to the immune system any antigen of interest in 10 copies, 5 copies at this size of the protein, and 5 copies at this size of side of the protein. So this is a model of a given antigen that can be presented to the immune system in this highly ordered and high number of copies to the immune system. What are the advantages of this technology? We were able to protect this. is an innovative delivery system. We think that has some optimal antigen presentation to the immune system and intrinsic adjuvant properties. And one of the main advantages is that we can use this system to present a small molecules like polysaccharides and peptides, but also protein domains and full proteins. In most of the cases, VLPs are not able to present protein domains because there is an steric hindrance and a folding protein that cause problems for production of these protein particles. In the case of VLS, being a particle of medium size, only 180 kDa, and having this particular structure, we think that give us a window of opportunity for presenting to the immune system biggest antigens, and in the native conformation that is crucial for neutralizing antibodies in many applications. These are the different competitive advantages I already mentioned. What we did at the lab also was we learned how to disassemble and assemble this protein particle. We can, for example, go from the chimeric structure to a folded pentameric structure, and then to unfolded monomer, and we can do this going farther and going back. So this can be used, this type of equilibria can be used to associate and reassociate the protein particle, for example, to create mixed chimeras that will present two or more different antigens to the immune system. An important feature is that protein is very resistant to urea. So you see here ACD, the protein in plain buffer, and in urea 8 molar keeps completely the secondary structure and resists also to high temperatures. The protein only aggregate at almost 90 degrees. These are all features that are very interesting for protein engineering. What are our coupling strategies? Essentially, we use two different coupling strategies. One is that the one that I showed you before, that is the genetic fusion. We just fuse at the end terminus the open reading frame of the antigen of interest, and then we produce the protein decorated with the antigen. But we also have developed another coupling strategy using high affinity heterodimeric peptides. These are low peptides that came from the leucine zipper and have very strong affinity constant between the heterodimeric peptides. So we decorate our particle with a peptide of about 14 kDa, and we put the other peptide in the antigen of interest, and when then we can mix in vivo and produce this type of particles. So using these two different coupling strategies, we were able to demonstrate that we can create particles decorated with small protein domains like this 9 kDa RNA binding domain, to different binding domains with different activities and different topology from alpha and beta structures. And we were able to demonstrate that this protein can be produced and can be present the antigen to the immune system in a very immunogenic setting. We also demonstrated that we can use these leucine zipper adapters to colocalize different functions in a given place. For example, here we created a chimera that is harboring BPA, that is a protein from rotavirus that has sialic acid binding activity, and GFP. And you can see here that when we mix both proteins in this kind of structures, we can have fluorescence in the membrane of cells because of the sialic acid binding capacity of the BP8 and the fluorescence from GFP. And this is the control where no BP8 is present. So this is a proof of concept that we can use this chimeric scaffold to localize in a given site with high ability different protein for different functions. And this this is a very short presentation of what we are doing. at Immunova, that is a startup company that was created based in this technology. These are all applications in veterinary medicine that we are trying to create a vaccine for bovine brucellosis. We are doing field studies with different prototypes of the vaccine. We are working also in foot and mouth disease. And these are all projects that are for human medicine that are under development right now. So finally, I would like to present the people that work in this project, especially at my lab, Paula Berger, that did all the immunological studies of BLS, and Sebastian Klinke, that did all the crystallographic structures, and Immunova, that is a startup, especially Vanessa Silverman, present here, that made all the structure and conformational studies of BLS and developed most of the technology, and other people working in the lab. And finally, people that supported our work, CONICET and the Ministry of Science Argentina, that give us a strong support, especially in the adventure to go from the lab to a startup company, and international agencies like Howard Hughes and John Simon Guggenheim that give us support for many years. Thank you.
Okay, we have time for questions. So I have one question, Fernando. During the immune response, the stability of this structure is conserved when you use it.
This is a very good question. We know that this protein is very, very resistant to proteases. But
in terms of immunogenicity?
In terms of immunogenicity, what we are trying to find out, and that's the reason we are doing experiments in cells, is the main question is how a protein given insoluble, for example, and secreted in a mice, can sustain for 2 years an antibody response in a mice. So what we think is that the protein is kept in some structure in cells to be presented continuously to the immune system. But we don't know where and what is the kind of the structure. In fact, nobody knows, but most people think that there are in antigen-presented cells, there are such a type of structure that can in some way accumulate proteins. And the resistance to proteases is a key aspect in this relationship with immunogenicity. For example, dendritic cells are very poor in proteases because they need to present for a long time proteins to the immune system. But we don't have a clear answer to this aspect.
Another question? Sorry.
The question is if when we give these antigens, if the animals are protected for the infection.